In the arena of Long Branch Falls Park comes the 75th Hunger Games.
Yo. Me. Me.
Hello and welcome to Capital News.
And I'm your host, Caesar Flickerman.
And I'm your co-host, Sunny Rose.
In honor of the 75th Hunger Games, the Capitol is excited to show an exclusive behind-the-scenes of all the work required to create the spectacular Hunger Games.
Let's go back to the very beginning. Today on our program, we have two Capitol scientists that set the precedent for all others.
They used their vast knowledge of recombinant DNA to create the event that we all love. Let's take a look.
Hello, my name is Shimmer.
And I'm Simmer.
We are the two chief scientists of the Capitol.
We are in charge of genetically engineering dangerous organisms to be used as obstacles in the games.
We bring entertainment to the show to spice up the tribute experiences in the arena.
Today we will be discussing our top secret techniques for concocting our most prized mutants that the tributes must face.
Based on votes from viewers at home, let's discover what was selected as the most popular and dangerous organism of the recent Hunger Games. Sting!
The clip that was just shown shows all the damage that these tracker jackers can do to the body.
But what makes these wasps so dangerous?
Let's go to the scientists.
Thanks, Caesar and Sunny.
Our genetic engineering process begins with a small, circular, self-replicating piece of DNA known as a plasmid.
First, we isolate the gene of interest, in this case, the gene of the venom of the poisonous snake, and its cloning vector, which is the plasmid. Then we introduce a restriction enzyme to both the DNA sequence and the plasmid. This cleaves DNA molecules at a limited number of locations.
Please. This process creates a restriction fragment with sticky ends at both sides. Sticky ends are single-stranded DNA, and the restriction enzyme creates complementary sticky ends in the plasmid and in the restriction fragment so that they can create hydrogen bonds and bond to one another temporarily before the DNA ligase goes over it and creates a more permanent bond. Now look. Okay. Also, the sticky ends are palindromes, meaning they are the same forwards and backwards.
In order to amplify the gene responsible for coding for the protein that produce poison, we must use polymerase chain reaction, also known as PCR. This technique requires incubation of a gene, as well as DNA polymerase, primers, and nucleotides.
Alright, next we insert the gene into the host cell. This is made competent by using cold calcium chloride and heat shock. This makes the membrane permeable to allow the plasmid DNA to enter the cell. However, only a few of the cells will incorporate the DNA into the genome of the wasp.
The stem cells of the wasp will contain the poisonous gene of the snake venom. When the offspring is produced, they will be the dangerous transgenic organisms that everyone in the games fear.
Wow, how interesting, especially since tracker jackers look like normal wasps.
Well, except for their larger size and golden coloration, and the fact that their venom was specifically engineered to target the part of the brain that generates fear, creating terrifying hallucinations that can drive a person crazy.
Well, to end our show, we're going to provide a sneak peek into the future games, which we thought would be pretty fun. Here's our specialist, Timmer.
That would be me. Timmer's my name, science is my game.
You go, Glen Coco.
He doesn't even go here!
Against the Capitol's wishes and share with you one of the most dangerous monsters we'll have on our show. I've created, using technology that Shimmer and Simmer talked about earlier, I created the Dinocorn.
What's a dinocorn? It's making me hungry.
It has nothing to do with popcorn. The Dinocorn is a cross between a dinosaur and a unicorn, and it has flying, super strength, and it can stab people with its horn.
It sounds really dangerous.
Competitors need to watch out because it's going to be the most unpredictable and dangerous creature out there. Using gel electrophoresis, we found restriction sites in the dinosaur DNA. Using endonucleases, we broke up the dinosaur DNA, attached it to the unicorn DNA, inserted it back into its mother, and we got the Dinocorn.
Intense. Now we'll watch a clip of the Dinocorn in the Capitol Laboratory.
The Dinocorn in its natural habitat.
Dinocorn has turned against his creator.
Where'd it go? There it is.
Run! Run!
Watch out for its horn!
Don't worry, folks. No one was hurt in the creation of that video.
Except for the 3 people that were killed.
Right, well, that's all the time we have for tonight's show. From Capital News at 11, have a great night and make sure to stay tuned for tomorrow's news on Cornucopias.
Go, go, go!
Bees in the trap, bees in the trap.
Bees in the trap.
Oh,
In
yeah,
the trap, bees in the trap.
I like
How are the tributes gonna deal with that? Watch out for the board.
Vote based on votes from viewers.
Make that face again.
No.
Make that face again.
It's up to record,
The Dinocorn has been spotted in his natural habitat.
Move weight himself, but living Hoboken. I spit that crap like I'm in that trap. So if you need a hit, then I'm with that bag.